inovasi, wujudkan impian. Kampus terbaik, integritas, kuatkan tujuan. Kampus terbaik, beri terbaik untuk negeri, untuk dunia, umat manusia dan alam semesta. Mari bersama hadirkan masa depan. Dalam langkah dan pikiran agar selalu terdepan, berbekal ilmu dan jiwa pengabdian. Deras arus zaman takkan tertahan, majulah melangkah, oh majulah. Kampus terbaik, inspirasi, tebarkan harapan. Kampus terbaik, inovasi, wujudkan impian. Kampus terbaik, integritas, kuatkan tujuan. Kampus terbaik, beri terbaik untuk negeri, untuk dunia, umat manusia dan alam semesta. Terus berpacu dalam keunggulan, lincah pembelajar untuk kehidupan. Inilah semangat baru, tegak raih kesempatan, wujudkan kampus terbaik selamanya. Mari bersama hadirkan masa depan. Dalam langkah dan pikiran agar selalu terdepan, berbekal ilmu dan jiwa pengabdian. Deras arus zaman takkan tertahan, majulah melangkah. Mari bersama hadirkan masa depan. Hadirkan masa depan. Pikiran agar selalu kampus terbaik, inspirasi, tebarkan harapan. Kampus terbaik, inovasi, wujudkan impian. Kampus terbaik, integritas, kuatkan tujuan. Kampus terbaik, beri terbaik untuk negeri, untuk dunia, umat manusia dan alam semesta. Mari bersama hadirkan masa depan. Dalam langkah dan pikiran agar selalu terdepan, berbekal ilmu dan jiwa pengabdian. Deras arus zaman takkan tertahan, majulah melangkah, oh majulah. Kampus terbaik, inspirasi, tebarkan harapan. Kampus terbaik, inovasi, wujudkan impian. Kampus terbaik, integritas, kuatkan tujuan. maju dalam keunggulan. Lincah pembelajar untuk kehidupan. Inilah semangat baru, tekad raih kesempatan, wujudkan kampus terbaik selamanya. Mari bersama menghadirkan masa depan, dalam langkah dan pikiran agar selalu terdepan, berbekal ilmu dan jiwa pengabdian. Deras arus zaman takkan tertahan, majulah melangkah. Mari bersama menghadirkan masa depan, pikiran agar selalu
Kampus terbaik, inspirasi, tebarkan harapan. Kampus terbaik, inovasi, wujudkan impian. Kampus terbaik, integritas, kuatkan tujuan. Kampus terbaik, beri terbaik untuk negeri, untuk dunia, umat manusia dan alam semesta. Mari bersama hadirkan masa depan, dalam langkah dan pikiran agar selalu terdepan, berbekal ilmu dan jiwa pengabdian. Deras arus zaman takkan tertahan, majulah melangkah, oh majulah. Kampus terbaik, inspirasi, tebarkan harapan. Kampus terbaik, inovasi, wujudkan impian. Kampus terbaik, integritas, kuatkan tujuan. Kampus terbaik, beri terbaik untuk negeri, untuk dunia, umat manusia dan alam semesta. Mari bersama hadirkan masa depan, dalam langkah dan pikiran agar selalu ilmu dan jiwa pengabdian. Deras arus zaman takkan tertahan, majulah melangkah, oh majulah. Kampus terbaik, inspirasi, tebarkan harapan. Kampus terbaik, inovasi, wujudkan impian. Kampus terbaik, integritas, kuatkan tujuan. Kampus terbaik, beri terbaik untuk negeri, untuk dunia, umat manusia dan alam semesta. Mari bersama hadirkan masa depan, dalam langkah dan pikiran agar selalu terdepan, berbekal ilmu dan jiwa pengabdian. Deras arus zaman takkan tertahan, majulah melangkah, oh majulah. Kampus terbaik, inspirasi, tebarkan harapan. Kampus terbaik, inovasi, wujudkan impian. Kampus terbaik, integritas, kuatkan tujuan. Kampus terbaik, beri terbaik untuk negeri, untuk dunia, umat manusia dan alam semesta. Mari bersama hadirkan masa depan dalam langkah dan pikiran agar selalu terdepan, berbekal ilmu dan jiwa pengabdian. Deras arus zaman takkan tertahan, majulah melangkah, oh majulah. Kampus terbaik, inspirasi, tebarkan harapan. Kampus terbaik, inovasi, wujudkan impian. Kampus terbaik, integritas, kuatkan tujuan. Kampus terbaik, beri terbaik untuk negeri, untuk dunia, umat manusia dan alam semesta. Mari bersama hadirkan masa depan dalam langkah dan pikiran agar selalu terdepan, berbekal ilmu dan jiwa pengabdian. Deras arus zaman takkan tertahan, majulah melangkah, oh majulah.
Hello, everyone. Good morning, afternoon, or evening, wherever you are joining us from around the world. Welcome to the international webinar with Food Safety Day 2025, with the theme of this year is the Food Safety Science in Action. My name is Asef Firman, as a Food Safety Master student in IPB University, and I'm honored to be your host today. It is really inspiring to see so many experts, academics, and professionals gather for this crucial discussion, especially on the food safety topic. And thanks very much for being here. First, we would like to encourage everyone to turn on your camera so we can engage more. This section, there will be the Q&A. So later you can share the question through the Q&A. As started, let's pray together to take a moment to reflect and seek guidance. And I invite everyone to pray in accordance with their faith. The prayer is begin. Amen. Today, we will explore the groundbreaking discussion on the food safety and in the event of the celebrating of the World Food Safety Day, including the insights into the Food Safety Master Study Program in IPB University. Before we begin, we would like to extend our deepest gratitude to our distinguished guest speaker, Professor Nigel French from Massey University, to our beloved teachers in the Food Safety Master Program, and also for Makepa, Food Safety Master Student Forum, for making this event possible. And let's continue to hear our national anthem of Indonesia Raya. Thank you. So now we would like to hear the opening speech from Professor Dr. Eko Hari Purnomo, STP MSc, as our head of Department of Food Science and Technology in IPB University. So the time is yours, Pak Eko. I hope Professor Eko is here.
Yes, I am.
Okay, thank you, Professor.
Okay, so thank you very much, MC Mas Asep ya. Okay, very good morning, everyone. I think I would like to deliver the opening speech in Bahasa, if that's okay with every one of you, because I think most of the participants are Indonesian, but before that, I would like to welcome Professor Nigel Friends, and thank you very much for being available to be the speaker for the webinar series that we have. Bapak dan Ibu sekalian, selamat pagi. Assalamualaikum warahmatullahi wabarakatuh. Senang rasanya hari ini bisa bertemu dengan Ibu dan Bapak sekalian di acara webinar kita terkait dengan Hari Keamanan Pangan Sedunia tahun 2025. Dan seperti kita ketahui, topik untuk Hari Keamanan Pangan Sedunia tahun ini adalah terkait dengan Food Safety Science in Action. Sebelum saya berbicara banyak ataupun memberikan opening speech, pada kesempatan ini saya juga ingin memanfaatkan untuk menyampaikan selamat dan terima kasih kepada program studi keamanan pangan Departemen Ilmu dan Teknologi Pangan IPB. Terima kasih Ibu Kaprodi, Profesor Rati Dewanti, dan juga tentunya beserta tim yang telah bersama-sama dengan Formakepa mempersiapkan internasional webinar hari ini. Kita tahu bahwa peran dari sains di dalam keamanan pangan rasanya bukan hal yang baru, begitu ya. Dan semakin hari peran ini tentunya semakin besar, ya. Mulai dari proses perumusan standar, begitu ya, proses perumusan regulasi terkait dengan keamanan pangan, sampai dengan kepada pengolahannya itu sendiri, pengembangan berbagai jenis food additive yang tentunya diharapkan juga bisa mendukung upaya-upaya peningkatan keamanan pangan, gitu ya. Bahkan sampai ke riset-riset di bidang pengemasan yang juga tentunya semuanya rasa-rasanya kalau kita yang bergerak di bidang ilmu dan teknologi pangan itu selalu memperhatikan aspek keamanan sebagai salah satu aspek utama begitu ya. Selain tentunya juga nanti berkembang juga ke aspek mutu. Jadi eh, peran itu akan terus berkembang dan akan semakin signifikan ke depannya dan rasa-rasanya ini menjadi sangat timely gitu ya untuk menjadi topik Hari Keamanan Pangan Dunia tahun ini dan tentunya juga eh, menjadi sangat apa ya sangat penting bagi kita karena hari ini kita bisa mendapatkan narasumber yang sangat kompeten di bidang keamanan pangan. Besar Michael Friends, once again, thank you very much and welcome to this webinar. So we are very grateful that you are available and willing to share your expertise with not only our students but also participants from other institutions around Indonesia. And even I heard also there is a participant from US in this international webinar. Jadi dengan demikian tanpa berpanjang waktu saya ingin membuka secara resmi ya internasional webinar tahun ini dengan tema Food Safety Science in Action dan semoga memberikan manfaat untuk semua participants dan juga diberikan kelancaran di dalam pelaksananya. Demikian, selamat pagi. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Professor Eko, for sharing the insight and forming this event. And now we would like to invite Professor Dr. Insinyurati Dewanti Haryadi, MSc, as our head of Food Safety Master Program in IPB University. To Professor Rati, the time is yours, Bu.
Okay. Good morning. Can everyone see the slides?
Yes, we can see.
Okay. Baik. Good morning. First, thank you, moderator, MC. Thank you, Prof. Eko, that has opened this webinar. Allow me also thank you, Prof. Nigel Friends. Hi, Nigel. Thank you so much. And I would like to have like 5 minutes to introduce our study program. And since most of us are in talking in Bahasa Indonesia, I would like to present it in Bahasa Indonesia. Sekedar memperkenalkan diri kepada Bapak Ibu sekalian. Assalamualaikum warahmatullahi wabarakatuh. Ingin memperkenalkan kembali pada Bapak Ibu program studi yang memang masih baru 2 tahun ini berjalan di IPB. Program studi Strata 2 Magister Keamanan Pangan. Keamanan pangan ada definisinya dalam undang-undang kita dan kita semua menyepakati definisi ini. Kita tahu banyak permasalahan keamanan pangan yang masih terjadi. Mungkin ini yang agak baru akhir tahun lalu ketika ada masalah pangan jajanan impor yang menyebabkan keracunan. Kemudian catatan keracunan yang meskipun underestimated tapi selalu mencatat sejumlah keracunan terjadi. Dan recently akhir-akhir ini dengan adanya program baru ini memang kita melihat ada peningkatan berita paling tidak ya tentang keracunan pangan. Belum lagi permasalahan keamanan pangan yang terkait dengan insiden perdagangan seperti penolakan produk perikanan baik ke Eropa maupun di Amerika Serikat. Kemudian adanya bahaya-bahaya kimia yang dideteksi atau kadang-kadang hanya permasalahan standar yang berbeda sehingga kita perlu mengetahui regulasi yang berlaku di suatu negara ya sehingga hal yang mungkin diizinkan di satu tempat tapi tidak tempat lain. Permasalahan kebutuhan SDM di bidang keamanan pangan ini sudah kami teliti di tahun 2022 dan memang ada kesenjangan antara jumlah yang tersedia dengan kebutuhan yang ada. Ini adalah data-data yang kami peroleh dari PRCOM waktu itu Badan POM. kebutuhan yang cukup besar tentang tenaga pengawas. Kami juga melakukan survei di berbagai otoritas kompeten keamanan pangan lain yang umumnya memang membutuhkan tenaga yang memiliki kemampuan atau kompetensi di bidang ini, dan tentu saja industri yang selalu memerlukan quality assurance, regulatory affairs, dan asesor keamanan pangan. Nah, pendidikan keamanan pangan memang relatif lebih baru ketimbang ilmu pangan ya. Mungkin kita catat salah satu yang awal adalah di Wageningen University, banyak di
Mohon maaf,
uh,
Bu Rati, boleh
ya.
bantu di slideshow, Bu? Nampaknya
Oh,
masih double.
oh, oh gitu. How do I do this? Oke, hold on second. Gimana caranya?
Mungkin boleh ke display setting, Bu? Yang atas, boleh.
Kok saya nggak bisa ya? Sorry, karena tahu masalah laptop saya yang masih ini. Hah? Oke, kecil ya tadi ya?
Iya, sekarang kecil.
Oke. Sorry, karena saya ada sedikit masalah sebentar ya. Saya stop dulu ya. Oke, saya stop sebentar. Karena memang saya ada masalah sedikit di laptop saya.
Ya, terima kasih. Ya, baik, Ibu sudah.
Oke, sudah lebih baik kan?
Ya, sudah. Terima kasih.
Oke, saya akan lanjut ke tadi. Suara saya terdengar?
Yes, sudah terdengar, Ibu.
Baik. Oke, tadi saya baru sampai di sini ya, menunjukkan saja peta program-program keamanan pangan yang banyak atau diinisiasi di Eropa, mulai di Amerika Serikat, New Zealand, tempatnya Pak Nigel Farage, dan kemudian juga ada beberapa di Middle East dan di Asia Timur, dan kita pada tahun 2023 mengestablish program studi yang pertama kali dan satu-satunya saat ini di Indonesia yang memfokuskan diri pada keamanan pangan. Ini adalah profilnya untuk Bapak Ibu yang berminat, terutama yang ingin memperdalam, kita ingin menjadi program pendidikan bertaraf internasional dengan misi menghasilkan SDM yang baik dengan kemampuan untuk melakukan atau menyelesaikan permasalahan keamanan pangan yang berbasiskan ilmu dari PAS dengan moto atau semboyan dari Hari Keamanan Pangan Sedunia tahun ini, dan risk based ya, berbasiskan risiko, meningkatkan mutu peraturan dan kebijakan keamanan pangan serta meningkatkan mutu riset keamanan pangan. Oke, di sini profil dari lulusan. Biasanya sebelum kita menurunkan pada kurikulum, kita sudah punya profil lulusan apa yang ingin kita hasilkan dari program ini. Ini adalah kurikulum yang kita miliki. Bapak Ibu bisa melihat yang biru, kuning, dan kemudian biru muda itu adalah bekal-bekal utama bagi seorang master yang bidang keamanan pangan, ya, mata-mata kuliah apa yang harus mereka miliki, kemudian pengayaan baik dari program studi maupun dari luar program studi, ya, kita misalnya memiliki toksikologi, rantai pasok, dan halal, ya. Kemudian beberapa tugas akhir yang memang wajib di IPB. Okay. Sehingga in short, perjalanan seorang mahasiswa di PSN KPN dimulai dengan diterima dulu melalui tahap seleksi, kemudian mengikuti perkuliahan, mempresentasikan rencana penelitiannya dalam suatu kolokium, melakukan riset, kemudian menyeminarkan hasil, mempublikasikannya, ya biasanya sampai submit atau sampai di review, kemudian melakukan ujian, dan seharusnya dalam 4 semester bisa lulus. Ini adalah template yang sebetulnya sudah kita polakan ya. Tentu saja kadang-kadang bisa saja ada tidak tepat, tapi kita harapkan semua bisa menyelesaikannya dalam 8 semester. Oke. Program ini juga ingin punya dampak ya dalam SDGs. Kita tahu bahwa status kesehatan yang baik berawal dari asupan makan yang bergizi, akses terhadap pangan yang bergizi penting dari masyarakat, dan untuk bisa memanfaatkan seluruh aspek gizi tadi, maka keamanan pangan adalah aspek yang paling mendasar, paling fundamental. Pesan, kalau kita ingat pesan Hari Keamanan Pangan Dunia yang pertama, waktu itu adalah jika tidak aman, maka itu bukan pangan. Jadi kami berharap yang berminat bisa join us. Untuk info lebih lanjut, silakan menghubungi alamat email ini, dan mudah-mudahan kita bertemu di semester depan. Terima kasih. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Terima kasih. Thank you very much, Bu Rati. And sorry for everyone for the trouble shooting. I'm sharing my screen again. And you see here that every year, WHO is leading the celebration of Food Safety Day. And the theme of this year, it is about the food safety science in action. You see here that science provides the fundamentals basis for food safety. To know more, let's watch the official video of this year term from WHO and FAO.
Hello and welcome. Greetings from the Food and Agriculture Organization of the United Nations, FAO.
And greetings too from the World Health Organization, or WHO.
Every year on the 7th of June, FAO and WHO jointly lead the celebrations of World Food Safety Day.
Because no one should get sick from eating food, and everyone has a role to play in keeping it safe.
This year, we celebrate the science behind food safety with the theme Food Safety, Science in Action.
Science helps us to understand what makes food unsafe and guide us on how to prevent foodborne disease.
But it is not only the knowledge of science that keeps our food safe, it is rather our actions. Actions based on the guidance and advice developed by scientists.
Governments rely on science to inform their policies, and food businesses follow scientific advice to apply it to good production practices.
And consumers rely on science-backed advice from governments and food providers when they make decisions about the food they eat.
So science is at the heart of food safety.
On this World Food Safety Day, we acknowledge the work of scientists, educators, and the contribution we all make to food safety through our actions.
The use of scientific knowledge is key to reducing illness and related costs, and more importantly, it saves lives.
Join us this June the 7th for World Food Safety Day to celebrate our shared commitment to science and food safety.
Together, we can ensure that all people everywhere consume safe food.
Let's put science
in
into action.
Okay, so now we come to the main agenda with Dr. Valerina Armeta, STP MSc, as the lecturer in IPB University. She will be as the moderator of this event. And first, I'd like to share with you about the personal information. Dr. Valerina Armeta is the Bachelor of Agriculture Technology in IPB University, as well as the Master of Food Science, and also Doctor of Food Science, IPB University. And she is a lecturer of the Chemical Engineering Program, School of Engineering in IPB University, Department of Food Science and Technology. Without further ado, I would like to hand it over to the session to Bu Valerina. So the time is yours, Bu. Thank you.
Okay, thank you, Mas Asep. Good morning, ladies and gentlemen, Bapak Ibu yang kami hormati. On behalf of the organizing committee, it is my distinct honor to welcome you to this plenary session for the international webinar that held by our food safety program or food safety master program, to celebrate the World Food Safety Day 2025. As introduced by the committee, my name is Valerina Armeta. My college and students call me Dr. Val or Miss Valel, and I am a junior lecturer in the Department of Science and Technology. The committee now in this webinar give me the privilege to serving as your moderator today. Yeah, with this webinar, we hopefully becoming the center of transformative role of genomic sequencing technology in food safety, yeah, in Indonesia food safety. So we are delighted to be joined by our distinguished speaker, Prof. Nigel French. Welcome, Prof. French. Yeah. Before Prof. French hour of speaking, let me to introduce him. Prof. French is a leading expert and distinguished professor in the field of infectious disease epidemiology and public health microbiology at Massey University, New Zealand. And he is also emeritus director in the infectious disease research platform from Tangata Treaty of the Taniwa. With respect, I hope I didn't misspell it. Yeah, and he is also the Emeritus Director of the New Zealand Food Safety Science and Research Center. Yeah, Prof. French's research has significantly advanced the understanding of zoonosis, antimicrobial resistance, and pathogen genomics. And we think that Prof. French's field of research will advancing our Indonesian food safety. Yeah, so today in our international webinar on World Food Safety Day, of 2025, yeah, Prof. Franz will give the presentation, the talk about the application of next generation sequencing and whole genome sequencing for the foodborne disease investigation. And ladies and gentlemen, Bapak Ibu yang kami hormati, this webinar session will be delivered in 3 segments with a dedicated question and answer segments or period following the each talk, yeah, to promote our meaningful engagement. So please feel free to submit your question in the chat throughout the session because we will have 3 segments, as I mentioned before, and we will address your question in the duration of Q&A section. Yeah, I think we will have around 10 minutes until 15 minutes Q&A intervals. Yeah. Okay then. Okay, then let us begin. Yeah, we will begin with the first talk. In this first talk segment, Prof. French will provide an introduction to sequencing technologies. I mean, next generation sequencing and whole genome sequencing. Yeah, that outline the broad scope of their application. Yeah, both of NGS and WGS, and guide us on how whole genome sequencing data and next generation sequencing informatical basic data can be interpreted in the context of food safety. So in this first talk, the general definition and concept will be given by Prof. French. Now, without further ado, Prof. French, the time and screen is yours. Do you prefer to share your screen by yourself or?
Yeah, I'll do that.
Okay. The time is yours, Prof.
Okay. Can you see
Okay. We have seen the
full screen or are you seeing the slide?
Still both
Not yet.
screen. Can you swipe it to press to the
Yep.
slideshow?
Got that.
Okay. Okay,
Wait,
good.
Okay, great. Well, thanks very much, and thank you to Professor Rati and all the other people that have been involved in organising this this wonderful occasion. It's a really important week, and it's a very important topic, particularly around the area of science in action, as was mentioned there, the advocacy by the WHO and FAO. And it's something in my career to date has been extremely important to me. Now, I work at a university and have worked at Massey now for over 20 years, but I have also worked very closely with the ministries of health and the Ministry of Primary Industry, which is the food safety regulator in New Zealand. And so I've had the privilege of being able to work up closely on using science to directly inform decision making at a government level as well. And I think that's been a really important driver for me. And what I hope to do over the few sessions now is at the very least to enthuse everybody that's participating in this talk about the importance of whole genome sequencing and the potential that it has to particularly inform decision making in food safety at government level, and also as a research topic for academics as well. So I'm going to divide the talk into 3 sessions, as was mentioned. The first one is a talk of around about 30 minutes, and it'll cover the sequencing technology, the applications, and how to interpret whole genome sequencing in a food safety context. Then we'll have a 10-minute break, and then I'll do another talk where I will specifically focus on examples to illustrate what I just mentioned. And many of those are examples that I've been directly involved with, so I know about them in detail and can answer any questions that may come up from that. And then I'm going to talk about the publicly available tools that many of you may want to use, or some of them, to interpret and visualize genome sequencing data. And then finally, after another break and a few questions, hopefully, we'll talk about some of the issues, a short talk on standardization, quality control, coordination, and accreditation. So some of those aspects there. So it'll be in 3 parts. And as I say, I'd be really keen to have some questions. Some of the questions I may obviously answer in subsequent sessions, but hopefully there'll be plenty of things to talk about. So firstly, whole genome sequencing, what is it? It's, if you think about in most of the pathogens that we deal with in a food safety context, bacterial pathogens, it's sequencing the entire DNA, potentially RNA content within the cell. So that could be the bacterial chromosome and or plasmids that are present within the cell. So you get the entire genetic blueprint through this process of whole genome sequencing. Any of you who've been involved in using earlier methods, some such as whole genome-based methods, pulsed field gel electrophoresis, you'll see that the genome sequencing has a number of advantages over those earlier processes. It's much higher resolution, it's more reproducible, it has many, many advantages over some of those earlier technologies. And since 2010 and the advent of the next generation sequencing revolution, we've seen a number of technologies emerge and become dominant in the field. And if you have genome sequencing capability, it's likely to be either an Illumina, and there are many different machines that produce Illumina, which are short read sequencing technologies. Hopefully you'll become aware of what I mean by that, or Oxford Nanopore technology. Those are the 2 that I know, certainly in my area, that are being used most commonly. There's also another long read sequencing technology called PacBio, which I have used, but mainly we use Illumina and Oxford Nanopore technology. And that little device on the right-hand side of the screen is an Oxford Nanopore MinION. It's a very small piece of equipment, but very, very useful. So what can you do with whole genome sequencing? You can, at high level, you can determine what the species of the organism is that you're looking at. You can also backwards calculate what the serotype. So thinking of Salmonella, for example, you might be able to determine what serotype it is. You can look at single nucleotide changes. So these are the bases ACGT, and looking at single changes within those genomes. Or you can scale up to a locus within a gene and look at a particular unique sequence, which is also called an allele. And there are basically these 2 approaches, the SNP or single nucleotide changes, and multi-locus sequence timing or allele-based methods. And hopefully that will become clear later as well. You can also look at genes associated with antimicrobial resistance. Now that could be antibiotics, such as penicillin, 2nd generation, quinolones, or it could be sanitizers, such as quaternary ammonium compounds. You can also look at virulence genes. So all of this is contained within the genome. So this is another major advantage over some of these early technologies. And then you can look at the relatedness of isolates that you might have from a food sample, say from a clinical human sample, and to try and determine what the source of an infection might be. Basically, genomics looks at the genome content and often is based on single isolates. You extract the DNA from those cells, or you can use non-culture based methods, which I'm not going to talk about in any detail, but these are becoming more and more prominent, the so-called metagenomics field. And this is basically what happens with whole genome sequencing. And this is taken from a CDC picture. You've got your bacterial culture, you extract the DNA, you amplify it, you shear the DNA into little parts, then you amplify it, then you put it into your sequencing machine, and you get these reads, which are basically amplified sequences that are determined by the genome sequencing machine. And they can be short reads, can be several hundred base pairs long, or they can be long reads, which can be thousands of base pairs long. There are broadly two different ways at which you can assemble those reads to give you information about that genome. So you have basically the raw read data, and each of those reads may overlap, and you use technologies to try and then determine what the underlying sequence is that generated those reads. And you can use de novo assembly, which is assembling those genomes without a reference, or you can use reference-guided assembly or reference-guided mapping, and you basically end up with contigs with the de novo assembler, which are multiple longer continuous sequences, or you can map that against a reference and end up with a consensus sequence relative to your original genome. And then you do your further analysis, you do quality control, you can then determine a whole range of different features which you then use in your analysis to help with your decision making. This is a useful guide from the WHO 2018, is a publication, a landscape paper. Basically, you start with your raw reads, which come out of your sequencing machine, you do your quality control, then you can assemble it using de novo or against a scaffold or against your known genome, and then you can do a whole range of different analyses. But importantly in this slide, and what they point out here on the left-hand side, is the importance of epidemiological information. Genome sequences on their own have limited value, but they have enormous value when they're combined with other information, particularly epidemiological information, if you're looking at an outbreak situation, for example, and hopefully, again, that will become very clear. I'm going to talk a little bit about quality control in the final session, but just to introduce this now, there are a number of ways that you can assess the quality of your assemblies. And this is a really important step before you start interpreting the genome sequencing data. Read depth, which is the number of reads that cover each base in your genome, the coverage of the entire genome, so what proportion of the whole genome is actually covered. The number of contigs in your assembled genome, the fewer the better of those. And then there's these two metrics, N50 and L50, which you might come across. And basically, if you align all of your contigs up, starting with the biggest and ending with the smallest, the N50 is the size of the contig at which you've covered 50% of the genome. And L50 is the number of contigs needed to cover 50% of those genomes as well. So these are all metrics that you'd use in that initial quality control to try and determine how good your data is and whether it's of sufficient quality to help with your subsequent analysis and decision making. And this is a very important step. What do we use genome sequencing for? First of all, is just one of the reasons why genome sequencing is becoming more popular is that the cost per genome has declined. It's kind of leveled off a bit now, but as new technologies come on board, more and more people are doing it. The cost of doing whole genome sequencing of individual isolates has come down quite dramatically. In New Zealand at the moment, we're just genome sequencing some Yersinia enterocolytica isolates, and it's costing around about $150 US to do an isolate. And that's pretty much comparable with some of the other earlier methods that were being used. Now, I'm going to give some examples of how we use it. So in outbreak investigation, for surveillance purposes, I'm going to talk about source attribution. This is an area that I've been working on quite a bit over the last 20 years, which is basically saying, what is the host origin of human illnesses? So does it come from chickens? Does it come from cows? And then helping you to determine the precise nature of the origin of particularly of human cases and sporadic cases. Source tracking along the production chain. I'll talk a bit about that as well. And you can also use it to estimate when a pathogen, a novel pathogen, arrived in your country. And also thinking about it at a smaller scale, when a new pathogen arrived into a farm, a food factory, or into a food processing environment. So there are many applications. And the data that you have as well, you can also use the sequence data to develop PCR targets for diagnostic test development. That could be anything from isothermic methods to conventional PCR. It's very useful to have those genome sequences. And there have been some initiatives. This is an IBM Mars initiative on the right-hand side, looking at trying to establish what is the normal and abnormal flora within a supply chain and seeing if you can use that to determine when things have gone wrong. And also, lastly on there, helping to identify new interventions. If your genome sequence identifies resistance to a particular sanitizer that you're using in your production environment, for example, you may want to develop a new one or to apply a different approach in order to control that particular pathogen. I'm going to come back again to this later on as well. The number of available genome sequences that you can use to compare any sequence that you generate in your laboratory has increased enormously over the last few years to the millions of genomes. And millions of high quality genomes are now available from multiple foodborne pathogens all around the world. And this is just some examples of databases that are available. Genome Tracker, Global Microbial Identifier, the PubMLST database, and the NIH NCBI database. So these are ones that if you're using your genome sequencing and comparing it with other isolates, these are the kinds of databases that you'd be downloading sequence data from and using it to compare with your data. This is a slide from a recent review in Nature Reviews Microbiology that myself and my colleagues published, illustrating some of the genomic tools used for foodborne pathogens. And I'd recommend having a look at this paper if you're interested in this area. We can combine genomics with modeling and epidemiology, and we can look at things like transmission pathways. So who acquired infection from what source and from whom? What's the attribution in terms of the animal reservoirs contributing to a foodborne illness? Where has my pathogen come from in the world? And that's where the value of those global databases come from. When did it come into the country? How fast is it transmitting? And also, how has it evolved over time? And this is more than just outbreak investigation. You can use it to inform public health policy and food safety control measures if you can understand what are the determinants of the propagation of pathogens right the way along a food chain. A word about whole genome sequencing and outbreak investigation. This is from the CDC a number of years back. When they started to deploy whole genome sequencing, they made an observation that compared to previous methods, more foodborne outbreaks or more outbreaks in general were being solved with fewer cases per outbreak. And this was attributable to the use of whole genome sequencing and improving the general efficiency of public health. Again, this is another figure from that review paper that I mentioned. Previously to genome sequencing, we'd have a situation at the back there where you'd have a low resolution typing method, and you'd be linking cases that really shouldn't be linked. And that would not only give you a higher perceived and larger and longer outbreak, but you wouldn't be able to tell that actually it's comprised of different outbreaks. And if you were able to identify each of those outbreaks individually, it's quite likely, be able to resolve those outbreaks sooner, resulting in fewer cases. So this kind of underlines the importance and value of genome sequencing. And there are some really, really good examples. And as I'm not going to be able to give you a comprehensive view of the application of whole genome sequencing in food safety, I'm going to be pointing to a number of key publications that, if you're interested, you can see how it was done and the value of these approaches. And these are two examples. And one of the earliest ones was the outbreak of this novel STEC strain, sugar toxin E. coli, that emerged in Europe, and the methods that were used there. And also a bit more recently was the outbreak, the huge outbreak of listeriosis in South Africa associated with processed meat. I'm just going to spend just a few minutes just talking about, in broad terms, how we interpret whole genome sequencing data, just so that you can get a feel for the nature of the data that's generated and some of the analyses that we do. In principle, I'm using made-up examples, and then I'll use some real examples later to illustrate how we do them, but just to illustrate some of the key points. So there are broadly, as I mentioned, two different ways of analyzing differences between genomes. And this is really important if you're wanting to compare an outbreak strain in a food with human clinical cases, for example. One of those approaches is the single nucleotide polymorphism, or single base changes. As bacteria replicate or viruses replicate, mistakes in those genomes occur, and those mistakes can be picked up as these so-called SNPs within genes and within loci within genes. An alternative approach is the multi-locus sequence typing approach, where instead of SNPs, your unit of interest is your allele. So if you look at the diagram in the top right there, you have a whole genome here, and within that genome you have genes, and within that you can identify a locus within a gene. And each of those locus or loci have sequences, and you may have SNPs within those sequences. A unique sequence for an individual locus is given a number as an allele, and those alleles are then combined to give you an allele profile, which is the unit of what you use for in your analysis. And broadly speaking, allele-based approaches are better for certain types of organisms. So things like Campylobacter, where they recombine and whole large chunks of DNA are exchanged between bacteria through different mechanisms. Allele-based approaches generally are better than SNP-based approaches. Just to illustrate this with a made-up example, but hopefully you can see. So this is an example taken and inspired by a publication on listeria in ice cream. So I want you to imagine that you have an ancestral sequence, which is the original source of all the descendant strains that you're interested of your organism, in this case listeria. And this is just a part of that sequence, but you can imagine it was the whole sequence. And I've just highlighted their bases in this original sequence, which could be in wildlife, it could be any other source, that are going to change as I show this example. Now imagine that sequence then contaminates the area or the environment of individual food suppliers. So these could be, for example, ice cream factories. 4 different suppliers there. And early on, we could see that they've already had a base change, that C to G has changed prior to it infecting supplier A and supplier B. Supplier C and D have the original sequence. Now, as the organism replicates within those suppliers, further changes occur. And those further changes are a result of those mistakes that are made, and some of those bases or the variant sites there you can see that are changing. So in supplier A, the A goes to a T, the supplier B, the C goes to a G, and so on. Now you can extract just those variant sites to give you what's called a SNP profile for each of those. And each of those show the bases that have changed from the original strain as they've evolved through those individual suppliers. And using that information, you can then, if you have the data from your final product, so your product that you might be interested in, what the source of that final product was, you can then create what's called a distance matrix. The distance matrix just tells you how many SNPs different there are between every pair of these. So there are 2 SNPs different between supplier A and supplier B. So you can see that they vary at the 3rd position and the 4th position, the T and the C. And likewise, you can do that for every pairwise combination. And then often what we do is draw a tree. This is a very simple tree, and you can see from this tree, that distance there is a single nucleotide change, that between supplier C and supplier D is 1 + 1 SNP. So we can see supplier C and supplier D in the matrix here. There are 2 SNPs difference. Between the final product and supplier A, there are no SNP differences. And likewise, between the other, if you add up the horizontal distances, they add up to the number of SNP differences here. And what you may want to be able to conclude from that, particularly if you have epidemiological information, is that the source of contamination of the final product is most likely to be supplier B because it has the fewest number of changes. So that's broadly speaking how you would interpret a SNP-based analysis. Now, multi-locus sequence typing, there are many, many different schemes. So some schemes, the earlier schemes, this is the allele-based approach, just use 7 genes. And now we have full genome sequences. We often have what's called the core genome, which is the number of genes that are common to most, if not all, of the Listeria in this example that you have, maybe around 1,700 genes. Or you can have a whole genome scheme, which includes all the genes that are in all known Listeria, and that may be around about 4,000 genes. So you can have different schemes according to how high a resolution you want to go. Now, using that same approach, rather than SNPs now, we're looking at alleles or unique sequences with individual genes or loci. So we've got 4 genes along the top there, and each gene at that locus has a unique sequence. And you can see the gene A in supplier A, you've got the sequence, so that is given a number 1. Supplier B, it varies at that CG locus, so it's given number 2. Supplier C varies again, so it's given number 3. And supplier D and supplier 4 also vary, and so they're given, and the final product, so they're given a number 4. And you do that for all of the loci. You give it a number if it's a unique allele profile, and then you have the allele profiles for your suppliers. So we then go from that to a distance matrix in a similar way to what we did before, and from this you can conclude, in this case, supplier D is the most likely source because it has an identical allele profile to the final product. So here we're looking at alleles rather than SNPs. Now, if you wanted to add the SNP data to that, you'd see that there are quite a number of SNPs that vary, but in terms of alleles, it's much fewer. And this is very useful in the situation for an organism where you have recombination, which introduces a large number of SNPs with every evolutionary event. So it just is a different approach and often applied to different organisms. Now, very often you'll see trees, and there are many, many ways of displaying trees to show relatedness of isolates. And basically what we see here is the shorter the distance between the tips of the trees, the more closely they are related. And the way you show these trees can vary according to the software package you're using or the underlying method. So for example, B, the minimum spanning tree there, is very useful for allele-based data because it shows, in this case, you've got a particular sequence type with a unique allele profile. It then's connected to others according to the number of allele differences between them. And so this minimum spanning tree approach is often very useful if you have allele-based data. Whereas if you have SNP-based data, you might have a rooted horizontal tree. You might bend that round if you've got very large amounts of data into a circular tree, or you might use what's called a network tree, which is an unrooted tree. And so there are various different methods, and it depends on what you're trying to get from your data. The data in here are actually all from the same underlying data set. There are many ways of drawing trees. You don't have to draw trees. If you want to infer relatedness between isolates, there's an alternative method which is used, which is also known as the barcode or LIN code approach. And this is an example. So if you have a whole genome multi-locus sequence typing, so this is based on allele profiles, you can then interpret from right to left. If you have a profile here, which is the same in two isolates, you know it's identical at all alleles. If it's the same up to this point, it's identical up to 3 alleles, but varies at around about 3, between 3 and 0 alleles. If it's identical up to here, it's around 7 alleles different. So it's a non-tree-based way of directly comparing isolates according to their, in this case, allele similarities. And there's a paper on this, which was published in Molecular Biology and Evolution, which explains this approach. It's also called life identification numbers, or LIN-based methods, where you can use this barcoding approach rather than a tree-based approach to infer similarities between pairs of isolates. So hopefully you can see how that would be applicable in an outbreak type situation. How can we use that evidence to identify whether a pathogen in a human case has originated from another population? Or more broadly speaking, comparing pathogens from different populations, how do you know they've originated from the same source? And broadly speaking, there are 3 different methods so that you would combine. So one of them might be your SNP distance or your allele distance. And you might say, if you've got, say, fewer than 21 differences, then it's strongly supportive. If it's between 21 and 100, it's vague. And if it's more than 100, it doesn't support it. There's also what's known as bootstrap support, which I'll talk about in a second, and tree topology as well, which so these 3 things that you can use. Now, the first one, the SNP distance, this is taken from the same paper that I mentioned just previously, the Peitling paper. And it just shows how much those SNP differences can vary, even in known outbreaks. So an example here of an E. coli outbreak, the maximum SNP difference was only 4, so highly indicative of a similar source. Whereas some of these listeria ones, it's much higher. And we'll talk a little bit about why you might get higher SNP differences, even in the same outbreak, when we come to talking about types of outbreak a bit later. So the number of SNPs is one indication that you can use to infer whether a pathogen has come from a particular source. Bootstrap support is often used in combination with trees, and you'll see this often reported in the nodes of trees. And it basically tells you how confident you are in a particular branch of a tree. And the more confident you are, the more likely you would say that that tree branch actually exists. And that's very important when you then come on to determine the tree shape. Now, if you've got 100%, it means that no matter how often you permute the individual sequences within your data, that branch in the tree is always there. If there's uncertainty about the location of that branch, then that will be much lower than 100%. If it's less than 50%, it means that branch is only present in random permutations of that tree 50% of the time. So you're less confident that that branch actually exists. And I'm going to illustrate this with an example in a second. And then the last thing which I mentioned was the tree shape. And there are 3 different types of tree shape that you can use to infer origin. And one of them is a monophyletic tree, the green one there. So if say your human isolate and your isolate from your food product are on a single branch and there is nothing else there, that's called a monophyletic branch. If they're on a single branch, but there are other isolates from other sources there, then that's called a paraphyletic. And that's suggesting epidemiological link, but it's not as strong as a monophyletic link. And if they're on different branches, then that's called a polyphyletic tree shape, and they're unlikely to have come from the same origin. And just before I come on to some examples, just to stress, you really need to know your pathogen. So Campylobacter behaves very differently to Listeria, to Salmonella, to E. coli, to Yersinia. So knowing your pathogen is really important. They evolve at different rates, they mutate at different rates, they swap DNA with each other in different ways as well. So understanding your pathogen is key to interpretation of genome sequencing data. Broadly speaking, the mutation or substitution rate is around about in the area of 10 to the minus 6 per base per year. But this varies, this estimate varies according to the duration of your study, and it varies according to your pathogen. And this is a paper which you can have a look at, which shows how the substitution rates can vary between studies and between organisms as you sample over time. And the other thing is the importance of laboratory and computing errors. So even though you might have a SNP, it may have happened during culture of your organism. So it may be a false positive. And there may be other reasons such as sequencing error, which can also give you false positives in terms of the presence of SNPs. And that can also affect your allele characterization as well. And again, I'll come back to this later, but this is a paper that was published a while ago now, but it's still very, very relevant, talking about interpretation of SNPs in terms of true variation, false positives, and false negatives. Now, food safety applications and interpretation. I was going to finish this first section before we move into questions, just with some examples. And these are taken from that Peitling et al. Paper. So if you want to read that one in a bit more detail, it will give you some more information. So I'm going to talk about 3 examples. Identifying the source of an outbreak, matching food isolates from one company to environmental isolates from another company. So this is where one company might be supplying ingredients to another company. And the question is, where has the infection in my company come from in terms of those ingredient suppliers? And then the last one is about resident strains. Do you have a resident strain, say, of Listeria or Chronobacter in your plant, or is it a new introduction? So these are all useful questions that you can address with genome sequencing. As I mentioned earlier, whole genome sequence, one piece of information in the bigger picture. You have to consider all of these other aspects as well: epidemiology, traceback information, and other data. And you really need that information in combination with your genome sequencing to determine the most appropriate response. And the second point, bacteria with non-identical genome sequences can originate from the same source. So you might have a mixed, complicated source. So if it's a fecal source, for example, it could be hugely diverse. And likewise, identical genome sequence should not always lead to the presumption of causality. So you might get identical genome sequences and there actually be no epidemiological link between them. The goal of any decision maker using genome sequencing information is to combine that information with a clear knowledge of the complexities to help you determine the best public health decision to make. So the first one, identifying the source of an outbreak. This is an example from this paper. It's a flour outbreak of 0121, the sugar toxin E. coli in 2016. And just to show here, we've got isolates at the top here, which are not related to the outbreak. Then you have red labels are the human clinical isolates, green labels are the flour isolates. And you can see the bootstrap of that branch is 0.995, so very close to 1 or 100%, which indicates very strong support for that branch in the tree. So that's one piece of evidence that you'd say is indicative of a causal relationship. Secondly, the number of SNP differences is a maximum of 4 between any pair of isolates, and the average is around 1, so very small SNP differences between them. So again, that's supportive of a causal relationship. And thirdly, monophyletic. It's only the clinical isolates and the flour isolates that are in this branch of the tree. So it fulfills those 3 criteria. It fulfills the SNP criteria, the bootstrap criteria, and the monophyly criteria. The second example is the one where you might have an ingredient supply, multiple ingredient suppliers into your food factory, and you don't want to know where the isolates have come from. And this is another example, again, it's Listeria monocytogenes in ice cream. And you've got green isolates from the final product in your factory, and the blue labels are from environmental samples from one of your ingredient suppliers. And again, very high bootstrap support for that. It's a monophyletic branch of the tree. And although the SNPs are a bit larger, they're still relatively small. So this would support this blue ingredient supplier as being the source of the Listeria in your final product. And then the last example is around resident strains. So you may be interested to know whether, if you take a dairy factory, for example, a Chronobacter or Listeria, have you got a strain that's just lingering around in the environment and growing in the environment and continually contaminating your final product? And again, similar approach. The red label is a clinical example here, clinical isolate here, but these are samples taken over a 6-year period. And you can see again, strap of one, high support, monophyletic, and a relatively small number of SNPs over a long period of time. So this is strongly indicative of a resident strain. So that's finishing up with those initial examples. And just before we finish this session, so while you're thinking of any questions that you might want to ask, there's a Coursera course, if some of you may have done this already. And I'm realizing that as I'm talking to you, that many of you, this will be obvious and you'll have covered all this and you know it all, and it might just be confirming your knowledge about it. Some of you, it may be all relatively new. And hopefully what I'll do is, in the latter case, we'll give you plenty of examples that you can then look up and increase your knowledge of this particular area. And finally, I did write a chapter in this particular book called Whole Genome Sequencing for Food Safety. You may want to have a read of that. And that's the recent review that we did, which talks about genome-based approaches for foodborne bacterial pathogens. So that's finished that first session. So I'm happy to answer any questions. I might just keep the screen up for now because I'm worried about losing it before we move on to the next one. So are there any questions? If they're not, I'll just continue, but if there are, I'm more than happy to answer them at this stage.
Okay. Thank you, Prof. Fresh, for the insightful and comprehensive overview. Now we will open the floor to the question for first Q&A session. Ladies and gentlemen, please use the raise hand feature in Zoom if you want to speak directly, or you can type your question in the chat if you want to ask via chat. Anyone?
Valel, if nobody asks, may I?
Okay, yes, please.
But
Prof. Rati.
somebody else has questions. No?
Okay. I think there is no question,
Okay.
Prof. Rati. Go ahead. Go ahead.
Okay. Thank you, Nigel. It's very insightful. I think out of the three purposes that you mentioned, at the present, for example, in Indonesia, we are still looking at the first one, how to identify the source. And honestly, most has not been moving. I mean, we're still using a lot of the conventional, and maybe we tap some of the gene to check the matching, but it's not really the whole genome sequencing that currently been applied. So my question to you is, if we want to move to that direction, what is your suggestion? So should we start with a simple one like sequence and then using the either SNP or MLST, or just the direction? Because currently we have not gone to that step yet. Thank you.
Yeah, I think that's a very good question. Thank you. And I think the issue of how to get started, and particularly if you have limited resources. So if your access to sequencing technology is limited, the budget that you have to spend on these kinds of investigations is low, and you may not have the relevant expertise to be able to do the sequencing or to interpret the results, then my recommendation is to start small. And what I've often seen is people who want to get into this kind of area will start with something like a MinION technology. So the MinION, the Oxford Nanopore technology, the long read sequencing, the technology has really improved recently with the quality in terms of the accuracy of the base calling in those methods there. You could use a very focused approach. So if you have strong epidemiological information and you have some conventional typing methods which might indicate a source, what you can then do is just sample from those. So take a smaller number of isolates to then run through your genome sequencing to confirm or refute what you may have already concluded from your lower resolution typing methods. If you have something like Campylobacter, I would recommend using allele-based approaches. It's the same data, but you use allele-based approaches. If you have something like Salmonella, you might want to use a SNP-based approach. So a lot depends on those organisms, but the data that you have, you could use for either of those. So it doesn't really matter in a way, but just choose. I would particularly make sure that you go for allele-based approaches if you've got a recombinant organism like Campylobacter. So I think start small, and maybe an investment in the MinION technology. They're not that expensive compared to other technologies. They've improved enormously. There are loads of courses available to learn how to use them, and then apply them just on a small subset of isolates that you have where you know you've sampled them in a meaningful way, that's based on the information that you already have. And even with as few as, say, 5 isolates, you might be able to say, well, actually, this really does confirm that this human case has most likely come from this source. And it's just a confirming thing rather than a primary investigation.
Okay. Yes, thank you. Minion, I think we in some of the institution have had that equipment. So I think that's something that you might think is simpler than probably the whole sequencing, right?
Yeah.
Okay.
Yeah.
All
If you
right.
have access to Illumina technology as well, many people are now combining long read with short read sequencing. But some of the information I've had recently shows that so-called hybrid assemblies, where you've got long and short read sequences, are no better than the long read sequencing alone. So as long read sequencing is cheaper and you can often use it, I mean, it's the tools themselves are relatively cheap and you can use it in your own laboratory. It seems to be a good place to start.
Okay, thank you.
Okay, thank you, Prof. French and Prof. Ratih. Is there any question from the others from the floor? Okay, I think because there are no more questions, we move first to the talk to Prof. For the second segment. Yeah, the screen, Tom, is
Sorry,
yours,
hello,
Prof.
Bu
Oh yeah,
Valerina. There
oh
are
yeah.
some questions also in the chat.
Oh, okay,
Yeah.
sorry. There is
Okay.
2 questions in the chat, yeah. Okay.
Yes.
Prof. French, there is 2 questions. The first one from Devi in column chat. David asks you that what are the main challenges in analyzing NGS data for foodborne disease investigation and how can they be addressed? I think this one related to the next segment. Can you serve first, Prof?
Yeah, I think the challenges are, well, there are resource constraints. So having the technology in the first place, having the laboratory capacity to make sure that you isolate and you can do all the basic molecular biology that's needed to prepare samples for genome sequencing. That's a challenge. And also then the interpretation. So having good bioinformatics support to make sure that you can interpret the data appropriately as well. But there are a lot of tools that are available now which have made things a lot easier. And I'm going to describe some of those in the next session. But yes, it still is a challenge, but it's nowhere near as big a challenge as it was even 5 years ago. Things have improved enormously just recently around your ability to adopt these tools and technology. But it is a matter of having the physical resource, the capability, and also the personnel with the right experience to be able to do these things. And I think just one of the other issues, which I'll come on to in the last session, is around accreditation, data sharing, and things like confidentiality. And it becomes a real issue very often in food safety. If you're dealing with a public health setting, you have patient confidentiality. In a commercial setting, you have commercial sensitivity, and that can create all sorts of potential conflicts, which can be quite difficult to resolve, particularly if it's become clear that a food company, for example, has been responsible or has been implicated in a foodborne outbreak. And I know very good examples. So the South Africa listeria outbreak, the court cases are still going on from that. So even when you've done your analysis and come up with your decision and your intervention, there can be all sorts of downstream challenges dealing with the sensitivity around the use of these data.
Okay, Prof. There is still the challenges, but there is not impossible, yeah, Prof. Yeah. Okay, we go
Yeah.
to the next question from Desinta. Desinta, coming from the Food Safety System Certified Ice Cream Company, and they said that they always monitor the microbiological environment through the periodic swab test. But one day, Desinta and team found a positive Listeria monocytogenes in the production floor area. The question is there any limitation reference for Listeria monocytogenes for the environmental or production area? And how would we identify the potential root cause and how to prevent it from cross-contamination into the products? I think the question more general,
More general
besides
food
that,
safety
yeah.
question, and I think it's that is a general kind of sampling issue as well. So if you have a production environment, a lot depends on your regulatory system settings as well. So in New Zealand, we have very specific requirements around listeria in food products, in particular ready-to-eat food products. So a lot depends on your system. But if you're wanting to investigate a potential production environment source of listeria in your final product, then devising a sampling strategy which covers all of the key areas where you think you might have listeria in your environment, and looking to see whether you have resident strains that may have been there for some time. This would be the value of genome sequencing: is do you have resident strains that have been around for a long time that are basically colonizing your environment, or is it a new introduction? And if it's a new introduction, where has it come from? Has it come from an external source? So that would require you to take samples from an ingredient supplier or from the surrounding environment in order to infer where that has come from. But there are methods using genome sequencing which can really help with that approach, and I'll talk a little bit about that later.
Okay, so depending to our sampling method and
Yeah.
the regulation, yeah, Prof. Yeah. Okay. I think there is no more question for the first session. Okay, we can move to the second segment, Prof.
Okay,
The screen
great.
is yours.
Okay. So in this session, I'm going to talk about some very specific examples, and these are ones that I have worked on with my team here in New Zealand. So I'm very familiar with these examples. And one thing I would say just before I go on to this is I've worked on the application of genome sequencing now in New Zealand for a number of years, and food safety applications are one of the applications that we use this for. I also was leading the reporting on COVID-19, for example, where we used genome sequencing of COVID in a human pandemic situation. So these technologies are very transferable. And I've also used this for investigation of an animal health outbreak, which wasn't a foodborne pathogen, but it was an incursion that came in. And I'll use some of those examples as well. Really, the application of genome sequencing from global to local scales. So we can use genome sequencing to infer how pathogens have transferred around the world and when they moved from one continent or one country to another. And there's an example there of a paper, right the way down to how a pathogen has evolved within an individual human being. So those are the two extremes on there. Both the publications that my team worked on. The first is the global emergence of 0157 E. coli, and the second one is the evolution within a single person of Campylobacter following use of antimicrobials. So you can see the scale is quite different. So the first one I'm going to talk about is the global study. And this is using genome sequences that were for all around the world, from multiple different countries. And you use whole genome sequencing combined with the dates at which those isolates were obtained. So the graph on the left shows when human cases started to appear in the different countries that were sampled in this study. And then you can use what's called ancestral state reconstruction. So basically, you're going back through the tree to try and estimate when those internal points where those sequences converge happened in time. And so it's the date at which they have a common ancestor often correlates with when the pathogen entered a new environment. So those dates of common ancestor become extremely important when you're trying to look at timing and origin of the source of pathogens. And so this is a map from this paper that estimates the dates at which E. coli 0157 moved from one country to another. And it's estimating in the bottom right-hand corner, for example, that New Zealand had at least 3 different incursions, the first one in 1966, the last one in the year 2000. And we actually exported, the blue line is an export of E. coli 0157 from New Zealand to Europe around about 1997. And you can plot this on a movie map. So this is going right the way back through time, estimating when the first transmission events happened from Europe to other different continents, and right the way up to the 21st century when those movements happen. So that's just an example of how you can use genome sequencing and ancestral state construction to infer the common ancestors and the likely source of these pathogens. We've repeated that for New Zealand, and we find that most of these really important cattle or sheep pathogens, such as the Shiga toxin E. coli, and some of the other pathogens as well, including salmonellas, arrived around about the mid to late 20th century. And that coincides with when we had cattle imports coming from other countries around the world. So it's most likely that some of our most important pathogens came in with live cattle imports into New Zealand. So that's one example on the big scale of looking at global transmission. We can also look at a national scale. So I'm going to get right the way down to outbreaks in a minute as well. But national scale, this is another example from New Zealand of what we call a slow burn outbreak. And rather than an explosive outbreak, which might happen over a short period of time with a common source, you can sometimes get these slow burn outbreaks where you have an incursion of a pathogen and it spreads within the food supply and creates a number of cases which often go undetected before you get a chance to investigate it. This particular strain of Campylobacter jejuni was first detected in 2 human cases in 2014. And then it was found in 3 of the North Island. So New Zealand has 2 islands, North and South Island, for the 3 of the North Island poultry companies and the breeder flocks. And this was novel, this pathogen, because it was resistant to tetracyclines and fluoroquinolones. So it changed the profile of antimicrobial resistance in Campylobacter cases in New Zealand. We'd never seen anything like it before, so we know it most likely came from overseas. We don't know exactly where it came from, because it was quite different on that branch of the tree, very distinct from other strains. Again, using a tree-based approach, it was quite evident that it had been around a few years and had actually evolved into different lineages in the different poultry suppliers. So the red isolates are human isolates, the pale blue are one particular poultry company, and the yellow and the dark blue are another poultry company. So it was already beginning to evolve differently in those different companies. We were also able to look at the evolution of this strain, and we could see that it had a plasmid, and the plasmid most likely transferred the tetracycline resistance into the genome. So we can look in detail at that. And then we could use our knowledge of the genetic relatedness of these isolates, because it had always been evolving, and compare that with a number of features of the poultry industry. So this included things like the geographical and road distance networks between the individual farms that made up these different poultry companies. And we could look at a number of other epidemiological features to see what the most likely route and source of transmission of this strain was between farms and around our poultry supply. And to cut a long story short, that's described in that paper in Epidemics there. a long story short, it arrived just before the first cases were detected, around mid to late 2013. There was a lot of transmission within companies, but very strong evidence that there was local spread happening as well, and most likely through a road, potentially feed network as well. It was persisting within farms due to the use of partial depopulation and carryover of infection. And it was evident that all companies were contributing to infection. And we could also look at how it was evolving. So that's another example of the application of genome sequencing to a slow burn outbreak and how you can use it to see how it's transmitted around a poultry supply. Now, again, thinking in terms of a more localized outbreak and over a short period of time. So we use genome sequencing to identify types found in sources, and often you're trying to identify what the smoking gun is or where a particular strain came from. And you can use, as was mentioned earlier, low and high resolution methods, but the high resolution methods often help you resolve these more quickly. You can have, if you have an index case contamination, a simple source of contamination where all isolates from the source and the human cases are very, very similar, but that isn't always the case. And this is what's important to bear in mind. Sometimes if it's fecal contamination of a source from a disseminated source, you could have multiple different strains and even multiple different pathogens involved in an outbreak. So it's something to bear in mind. Going back to the example of the 104 STEC outbreak, just to illustrate another point, this was one of the first examples at which genome sequencing was used, and it was hailed as a really good model for how to do this in future. And this is the timeline over which events happened. It's quite a complex graph. I'm not going to talk about it in detail, but just highlight those areas A, B, and C. Now, B, starting with B, is the peak in the notified cases. So that's the peak in the onset date of diarrhea and the cases that occurred during this European outbreak, very large outbreak. Calculating back from that, knowing the incubation period, the peak in exposure was a few days before that. But by the time the source was identified, in this case, bean sprouts that were being eaten raw, and the public warning was issued, the outbreak was already over. So even though you can do all of this amazing tools, very often the outbreak's over by the time you get to understand what the source was. So that was just one important point. Now, Salmonella enteritidis is another example in New Zealand. We hadn't had an egg-borne Salmonella enteritidis outbreak in New Zealand beforehand. And so this was quite an unusual event. We had an Auckland restaurant in the North Island where we had 38 cases, 17 confirmed of this particular strain. And we wanted to know where it came from. It was most likely in a raw egg-based dessert, a tiramisu-based dessert. But there was obviously very strong interest, particularly it was an egg-based dessert, as where this particular strain might have come from. It was also detected through industry sampling and detected in breeder flocks as well. It was detected in some other animals, but they were considered most likely to be incidental. So the Salmonella enteritidis outbreak, the aims of the analysis were to understand and support those epidemiological investigations, so where human infections were coming from, estimate when it came into the country and where it was most likely to have come from, and also how it was being transmitted around the poultry industry. So those are the broad aims that we used. Just a quick result here. So if we look at the SNP distance between human cases and poultry isolates, many of them had a 0 SNP difference between them. And the most likely difference was just a single SNP. So this is strong evidence that the isolates from the poultry industry, from both broilers and from layer farms, were likely to be the source of those human cases. We did do some ancestral dating. So I mentioned this earlier. You might want to know whether this is a new strain that's just come into the country or whether it's been around for a long time. And one quick way of doing that is to plot the date at which your isolate was isolated. So this graph on the right-hand side, this is the date. So this might correspond to the date that isolate was isolated, this one to the date that isolate was isolated. And then you create a tree, and then you take the distance from the root of the tree to the tip of the tree, and you plot those two against each other. And this creates a correlation, and the intercept with the x-axis is your first indication of when that strain might have come into the country. So this was just before our Auckland restaurant outbreak. So this led us to believe that it hadn't been around in New Zealand for much longer than that. We conducted some more sophisticated analyses, which again confirmed this with some intervals around that. Most likely came into the country in the early part of 2019, prior to that restaurant outbreak. And basically, what it showed later on was that in those early days, prior to interventions in the poultry industry, most of those human cases came from egg farms, and then later on, mostly from broiler farms. So it had spread throughout the industry, both the meat and the egg layers, and both were contributing to human cases. Most of the interventions were in the egg industry, so the egg cases tended to disappear, and then we were left with ones mostly associated with the meat industry. Looking at, we're then able to compare our sequence type 11 genomes, such as the Salmonella enteritidis, with the global population that were available. So this is going to those international databases. There were 1,300 genomes available. We used a range of different techniques, so including the allele-based MLST approach, multi-locus sequence type. We built a distance matrix, a spanning tree, and then we looked at the clustering of those. And then we compared those with the New Zealand isolates. Now, this next plot looks very complicated, but this is those 1,300 genomes colored by their country of origin. So this is from the international database. And you can see it's quite a complex picture, but it's based on their similarity. So genomes that were very similar are closer together in this two-dimensional diagram. And our outbreak strain, known as CO1, is right in this little group here. And if we zoom in on that group, we find that the outbreak strain is closest to isolates from Europe. And in particular, there's one from the United Kingdom here, one from Turkey here, another one from the United Kingdom here. And this is another New Zealand traveller that actually travelled to Turkey. And these isolates from Turkey were in food products. So we can't conclusively, because it's not that close, but we can conclude that our strain most likely came from Europe, and we've begun to identify potential origins of the New Zealand strain. When we looked at how it's being transmitted through the industry, what became quite clear through our reconstruction of the genomes was that the hatchery or the breeder flocks were the most likely origin of this isolate. This particular strain came in and then got disseminated through chicks, through to the layer flocks, through to the broiler flocks. So the same hatchery transmitted that particular strain. Interventions were put in the industry, and this strain has largely disappeared. We think it may be still in some broiler flocks, but it's largely disappeared subsequently. And a lot of that decision to do something and how to do it was informed by the genome sequencing. Another example I'm going to give is another outbreak, a huge outbreak, which happened in a small town in New Zealand, in the North Island of New Zealand. The town is called Havelock North, and this isn't a foodborne outbreak, it's a waterborne outbreak, but it illustrates the value of whole genome sequencing. A huge number of cases happened in this particular region, and water was tested from the bore wells, from the water supply, and the taps. It was an untreated water supply, a groundwater supply, and the human cases, and Campylobacter jejuni was identified in those sources. So as a waterborne outbreak, over 8,000 cases. The population of the town was only twice that, so around about 50% of that population got sick. 4 people actually died as a result of this as well, and 42 were hospitalized. So this was the biggest instance of waterborne disease in New Zealand, and one of the biggest Campylobacter outbreaks in the world ever. It's written up in that nice publication there in Journal of Infection. This was the epidemic curve. Outbreak cases confirmed and probable. Once the water supply was chlorinated, the outbreak went away. So we knew it was the water supply. So the investigation then went on to, well, what contaminated the water supply? So samples were taken from the area around the wellheads. So there were a number of wellheads that were on a road that supplied the groundwater supply. There was a poultry farm up there. There was sheep grazing the area. There was a whole number of other things that were sampled. Whole genome sequencing was performed, allele-based methods. This is that minimum spanning tree showing the differences in alleles. You can see it's quite a complex outbreak with multiple different strains identified. But what we can see if we take this big group here, this particular sequence type, it was found in outbreak cases in green. Other outbreak ones that weren't linked but were in the same region, also in the reticulated water supply in the bore water and in sheep feces that were grazing on the top there. And you can see this strain also in sheep feces, human cases, the reticulated supply. So the conclusion from this, and what most likely happened, was sheep were contaminating the pastures around the wellheads. There was a huge rainfall event and sheep feces got washed into the wellheads. The electricity supply to the wellheads failed, and so the contamination of the groundwater happened as a result of direct contamination through that. People then drank the water and got sick. So it's a very good example of how you can use this to identify the most likely source of that infection. Now, we have a persistent burden of Campylobacter in New Zealand. And even after the Havelock North incident, which is this incident here, which caused a big spike in our notified cases, we still have this kind of plateau, which happened after interventions in the poultry industry. It's still persisting in a seasonal infection over time. Another way we used genome sequencing was using it for source attribution. And so this is the approach to looking at the source of sporadic cases. I've already mentioned how we use it to identify the source of outbreaks, but so-called sporadic cases. So these are the cases that just turn up routinely where there's no obvious source of them. And these can be quite complicated pathways. We basically have the human cases, and we then compare the isolates from those human cases with our potential source, which could be livestock, or it could be wildlife sources, or it could be environmental sources. So source attribution is a very useful way of identifying the source of infection. And it can be used to inform policy for prevention and control, so longer term policy as well as outbreaks. It can be used for surveillance, but also monitoring the impact of interventions that you might put in the food chain. And it's useful for sporadic cases and for outbreaks, but it does require this One Health approach, which hopefully you'll be familiar with, where you are looking at what's going on in animals, the environment, and in people at the same time. And I'm just going to give an example of how we look at source attribution. So with source attribution, you might want to think about what's most distal to the human infection, so the point of ingestion of the pathogen. And that might be your animal reservoir at the top here. So you might want to find out what the most likely animal reservoir is. Is it chickens? Is it cows? Is it sheep? Is it wildlife? Then you might want to say, well, what is the pathway that's being contaminated? And that might be food, but as I mentioned earlier, it could be water. And you might have a different regulatory authority responsible for food or water. So this might help you determine which agency is responsible for control. Then you can think, as you get nearer to the point of consumption, what the exposure might be. Is it milk, meat, drinking water, or is it recreational water? And then you might want to think of very specific risk factors. So you're getting nearer to the point of consumption. So we use these methods. And an example on the right, which is a food example, where you might have cattle as a reservoir contaminating the food chain, which is the pathway, and the hazard is in the milk supply, which is the exposure, and the specific risk factor is the consumption of raw milk. We can contrast that with a water example where you, as I mentioned in the previous example, you have sheep, which are the reservoir contaminating the water pathway. The hazard is in the drinking water, and the risk factor is the consumption of untreated drink water. So this is a framework for thinking about source attribution. We just conducted a study to try and understand what the sources of Campylobacter were in New Zealand. And this is a novel approach because it used genome sequencing to infer the most likely reservoir of human cases. And then we used a case control study for the different reservoir associated human cases. And we had over 600 cases, 600 controls, and we genome sequenced all of those isolates from the animal reservoirs and from the people, and then conducted a case control study. And kind of long story short, what we found was that poultry was still the most important source. No matter which way you looked at it, poultry was still the most important source, but cattle were also contributing to those human cases. When we look at our risk factors, we find with poultry-associated cases that chicken-associated risk factors are common. So consuming undercooked chicken, consuming chicken in restaurants came up as risk factors for all cases, but also for poultry cases in particular. Then when you look at the cattle-associated risk factors, you find risk factors such as raw milk consumption or living or working on a farm, particularly a dairy farm, becomes very important in terms of the risk. So that's a way of helping you refine your understanding of the source of those human cases. And then you can start, for example, in rural areas to think how you might put public health messaging out to try and reduce the risk associated with the consumption of raw or unpasteurized milk. So that particular study then identified poultry as predominantly causing urban sources. New public health goals were set working with the poultry industry, but we also had information to help identify what the potential rural sources were and put measures in place to try and reduce those risks as well. So just talking now, just shifting from talking about kind of national studies, so public health driven studies, to food processing environments. And many of the principles that I've spoken about already are highly applicable in these areas as well. You can use these techniques to micro map at plant scales. You can use it to understand how pathogens being transmitted and spread within food processing environments, identifying resident strains. All of those things can be used using those techniques. And in New Zealand, we've used this working with food companies for Campylobacter, Listeria, Chronobacter, and E. coli. And one of the tools that we've used is a tool called MicroReact, where you can map sequences according to where they are in the plant, and then you can look at potential parts of that phylogenetic tree. You can say, for example, this particular group on the bottom right-hand side of the tree there is located in a particular part of the environment, and that can help you identify where it might have moved from and to when you're looking at potential resident strains and the source of those strains. And there are some commercial systems that are becoming available. So Traxar, I think, is one. I've not really looked at this one in huge detail, but I think there's a lot of interest in using these techniques to inform sampling strategies. There's a question earlier about that, and the combination of that using genome sequences. And again, using those international pathogen sequencing databases can be really helpful in identifying the likely source of those infections. I'm just going to talk about some online tools that you might want to play with, as this came up earlier. Most of the tools that we use do require a high level of expertise in terms of bioinformatics, using command line tools, often using Unix rather than Windows operating systems. But there are more and more tools becoming available that are online that you can simply drop your genome sequencing data into and use those to make inferences. And I'll talk about Pathogen Watch in a second. The Centre for Genomic Epidemiology, that website there, has got a number of really useful tools that you can use with your assembly data, your contig data that you generate from your genome sequencing. And drop those in to these websites and it will produce some outputs, which is of value for you to look at. And this can include things like looking at antimicrobial resistance, virulence genes, serotyping, retrospective serotyping of based on the genome sequence. Pathogen Watch, I'd recommend having a play with this if you've got some genome sequencing data or you want to use some of the examples that they have online. This tool, you can basically drop your sequence data in the form of those assembled contigs. So you've got your raw reads, you then assemble those, and the assemblies create these FASTA files or FASTA files. You can then drop those into Pathogen Watch and it will help you then to do some basic analyses on these. So top right there example is a folder that I have with a number of genome sequences that have been assembled. You basically drop them into the Pathogen Watch site. It then does some analysis behind the scenes on those. It'll then tell you that you've managed to do some core genome multi-locus sequence typing, you've done some serotyping, and then you can do some further analysis by looking at those genomes. It's telling me that these are salmonella isolates that have got some sequence type 11 enteritidis strains in there, and also some sequence type 183 enteritidis strains in there as well. And then you can look in more detail at those, and it'll tell you what the multi-locus sequence typing is of each of those isolates. It'll do some in silico serotyping. It'll also tell you whether plasmids, virulence plasmids are there, and also enable you to draw a tree based on those assemblies. So it's a really good initial way. If you've got some data, playing around with it takes literally seconds for you to be able to get some inferences from your genome sequencing data. So it's a very useful tool. And it also gives you some genome sequencing quality metrics as well. I mentioned earlier about the N50 tool. This is one thing that you can also use to help you assess some of the quality of those genomes. Online tools for genomic epidemiology, MicroReact is certainly worth playing with. You can add maps of the genome sequences, you can look at phylogeny, you can add other data and have a timeline along the bottom. So this is a particular animal pathogen that I've mapped here along with the phylogenetic tree. This is actually bovine tuberculosis genomes. You can see the tree, you can see where each of the isolates are, and this is an interactive map. So you click on an isolate on the left-hand side, it'll identify it in the tree, and at the bottom as well, it'll tell you where it is on that timeline. So really useful tool for you to play around with, and lots of good examples. And the other example is NextStrain. NextStrain is a bit more difficult to set up, but we use this for our outbreak of Mycoplasma bovis. And used whole genome sequencing and put those isolates into NextStrain. And it's a very useful interactive tool for looking at genome sequences and for mapping and for helping you to look at how things are being spread around the country. And I'll just give you an example. So this is the Mycoplasma bovis. You can see those are outbreak farms on the right-hand side. You can see the phylogenetic tree on the left, and it also helps you to infer which farm has infected which farm. And you can actually play movies through time. So this is walking through that phylogeny and showing you where the most likely ancestral states were in which farms and how those different clades or strains emerged, where they were being transmitted from and to. Then the intervention took place, which was a culling-based intervention. And then gradually over time, several of those lineages disappeared, and we were left with one strain that was just in one focus within South Island. And then over time that disappeared, and we're now free of Mycoplasma bovis. So genome sequencing combined with these tools was very effective at helping to inform decision-making. And this is an example from COVID, but again, you can use these narratives, which can be very useful at transferring information in an interactive way to help with the interpretation of genome sequencing. And there's a paper there which describes this and a website as well. So that was the end of the second talk, probably just 5 minutes or so, if there are any questions. So what I focused on there was using some very specific examples, things that I'm very familiar with, and illustrating some of the tools that we use to inform decision-making by regulators, by industry, and some of the visualization tools, which are improving dramatically now to enable you to, any data that you generate, you can then very quickly do some preliminary analysis on those data.
Okay, Prof. There is a question in the chat from Pak Dede Yusuf. He asked, in your experience, where does WGS or whole genome sequencing provide more actionable insight during early monitoring of fresh produce and the cultivation and harvest stage, or in the surveillance of processed plant-based food products after packaging? And what are the distinct challenges and benefits in each stage, from the fresh, harvest stage, and surveillance of processed food?
So is that a question specifically about fresh produce? Was that the...
Yeah, well, from the
Yeah.
fresh product and
Oh,
the
okay, through to processing.
processed product.
Yeah. Well, this is where that review that I mentioned, the Nature Reviews microbiology review, talks about how genome sequencing can be used along a food chain. So if you think about the food chain going from the original source, so if you're talking about a fresh produce, it could be fresh vegetables that are contaminated with an animal reservoir. You're actually going back to what is the animal reservoir. And there's some really good examples in the literature of wildlife and animal reservoirs contaminating fresh produce, horticulture-based produce. So you can use genome sequencing at that level to identify what the source of contamination is. Then you can use genome sequencing throughout the processing chain to identify how those pathogens are being propagated through the chain. Is there an example? So questions you might want to ask is there a resident strain in your processing environment that's contaminating your product, or is it coming fresh from your product? Is there a strain that's being selected by your interventions? So if you are using, say, a sanitizer to clean your equipment, are you finding that your final product actually has a strain that's resistant to your sanitizer? And you can quite often infer that from the genome sequencing as well. So you may want to use that information to change your processes within your production environment to try and improve how you produce your food. So obviously you're limited by resource constraints around sampling, around the costs of doing things, around the expertise needed to do that. But certainly in New Zealand, we have some really good examples now where multiple industry players, and if you think about New Zealand, we export most of the food that we produce. So we're a huge exporter of food products to places like China and to Indonesia as well. So we export a lot of food. It's really important that our food is deemed safe. So our industries, from our horticulture industry, such as the kiwi fruit industry, is very interested in monitoring pathogens such as listeria and kiwi fruit. The dairy industry, in terms of Chronobacter contamination, for example, the poultry industry in terms of Salmonella and Campylobacter, is really good examples of how a whole food chain approach combined with genome sequencing can help with devising appropriate interventions along the food chain. So not just at the end, but actually early on in that process as well.
Okay, thank you, Prof. There is another one question from Dia Anaya. She said she find your presentation very insightful, but she have two questions. The first one is, how can we determine the speed of infection spread using NGS analysis? And the next, in outbreak investigation, when we do typically choose SNP over the MLST or vice versa. I think this one is monolocus sequencing. Okay.
Yeah. Well, perhaps taking the last question first of all,
Okay.
both SNP-based approaches and MLST are very useful, and very often you'll do both.
Oh.
And you might do core genome or whole genome MLST combined with SNP-based methods as well. But multi-locus sequence typing approaches are better for some organisms where they are highly, they have a lot of genome exchange. So organisms like Campylobacter, they are naturally competent organisms. They will uptake DNA from the environment through a process of recombination. They will also have bacteriophages, which transfer sequence data from one organism to another, as well as various other mechanisms by which genes can move in and out. Now, each of those individual events can transfer multiple SNPs through into the genome of those organisms. So that can create a distortion around the relatedness of pairs of isolates. So if you've had a recombination event, it will give you a false idea. If you just use a SNP-based approach, it will give you a false idea of how distantly related two organisms are. So for something like Campylobacter, you're probably better to use a multi-locus sequence typing approach because it's known as a panmictic or recombinant organism compared to more clonal organisms, and Mycobacterium being the most clonal, followed by organisms like Listeria and Salmonella and E. coli. So it depends on your organism. That's why you need to know what your organism is. I've forgotten the first part of the question now. What was the first part of the question?
Okay, I will re-mention it. How can we determine the speed of infection spread using the next generation or whole genome analysis? Is it possible?
The speed of spread.
Yeah, speed the spread.
Yeah. Yeah, so this is where this ancestral state reconstruction methods come in very useful. And I probably illustrate that best with our COVID examples. So COVID obviously being a virus, we use RNA sequencing for COVID. During the early stages of COVID, we were reconstructing the genome sequences of almost all the human cases that were coming into New Zealand. And from that, we could determine their date of common ancestor. And from that, based on the epidemiology and where they were, we could actually infer who gave infection to whom. So we might know that you have a family where you have multiple infections, and you want to know where that family got their source of infection from. And then you have other families that might go to the same faith-based organization, the same church or the same mosque, for example, and having sequences from there, you can then say, well, that family most likely picked up their infection from that community setting, or it could be from any other community-based setting. So you can use genome sequences based on the similarity and the dating to tell you when the infections occurred and when most likely that transmission happened. And using the dating, you can also say when big events happen. So you can say, well, this strain that came into New Zealand in 2022 in December, then moved from the North Island to the South Island in March of 2023. So you can do the dating, can tell you when things moved. And then based on that kind of information, you can also determine the reproduction number of the organism and say how rapidly it's been spread based on your understanding of the secondary transmission rate. So it's a bit involved, but we published quite a few papers on the application of genome sequencing for COVID. If you're interested in those papers, it's not foodborne pathogen, but it really gives you an insight into how you can use genome sequencing technology in the face of an outbreak where you're wanting to look at very specifically at transmission events and when they happened and how rapidly they're happening.
Okay, thank you, Prof. Because we have limited duration of the segment, I think we can move to the next talk session.
Yep.
Please,
Okay.
Prof.
This is a relatively short talk, this one, you'll be pleased to know. Sorry, I don't know why I jumped back then, but there we are. Oh, going all the way up, sorry. Right, so the last section. What I'm going to talk about here is some of the difficulties as well as the approaches towards standardization, accreditation, coordination, and quality control. So this is an area which there's a lot of interest in with the WHO, with FAO, with some of the other UN organizations, with food businesses, with regulatory authorities, because you've got a situation where you might have different laboratories using different techniques, and you might want to make comparisons. So how reliable can a comparison be between one laboratory setting and another laboratory setting? And for those of you who are familiar with things like pulse field gel electrophoresis, that was notoriously difficult. And that's where PulseNet came about, to try and standardize approaches and methods so that you could compare a banding from one pulse field gel with a band in one country with a banding from another pulse field gel in another country. And it was hugely difficult, and a whole lot of things came around to try and manage that. With whole genome sequencing, because of the way the data are generated, it's less of an issue, but it still is an issue. And so it's really important that we have good approaches to this. And we're not there yet, but there's a lot of effort, and it's worth keeping an eye out for how this is developing at a global level, but also at regional and national levels as well. So just a reminder that genome sequencing, if you get a SNP, it could be a true representative, a true variation that happened, and you can use that data to then infer transmission events. But you can also get false positives. So you might, in the process of culturing, generate SNPs in vitro. You might get mapping errors due to the way that the genomes are. So particularly if you have a lot of repeat sequences, then the mapping to and assembly methods can create errors. You can get sequencing errors as a result of your machine that you're using. You can get genes that are very similar to each other or of similar origin, and they can create mapping errors as well. You can get false negative too. So just you can get the absence of a SNP doesn't mean it's not there, and that can be due to, again, to sequencing errors and other things as well. So these things happen. What are the metrics that we use? There's a number of different approaches. So you might come across terms like coverage. So coverage is where you have the number of reads that are generated by your sequencer. You have the length of those reads, and you know what the size of the genome that you're sequencing is, or assuming that you know what the size of the genome is. And what you then do is you multiply the number of reads by the length of the reads and divide it by the size of the genome, and that gives you an average coverage of each base over that full genome. So the example there, if you've got a Salmonella genome, 4.5 million bases, you have 250 base pairs, 2 million reads, then you can calculate that every base on average is covered 111 times. So you have what's called 111x coverage. And that would be good. That would be good for a de novo assembler. If it's a lot less than that, particularly if it's fewer than 30, fewer than 10, then you'd be a little bit concerned. And poor quality areas need to be trimmed before you do that, but there are all sorts of tools for being able to do that. And you base that on things like what's called the Phred score, which is the probability of incorrect base calling, and that's related to the sequencer that you're using as well. You also have things like breadth of coverage. So that's the assembly size. So once you've assembled your genome divided by the target genome. So if you've got a genome of 5 million base pairs, but your estimate, your base assembly is only 4.5 million, then 4.5 divided by 5 multiplied by 100 gives you your breadth of coverage. So it's inferring that in this case, about 4.4% of your genome is missing in your assembly. And then you have sequencing depth, which is that at each base, so rather than the full length genome sequences, it's something that's a base specific thing. It gives you the number of reads at each base or each site. And if you're interested in particular at a particular part of the genome, you might be interested in the sequencing depth at a particular location. I mentioned earlier about N50 and L50. You'll come across these. So N50 is the length of the shortest contig needed to cover 50% of the assembly. So you've arranged your contigs in order of size, and it's that one that's the shortest one needed to cover that 50% of total size length. And L50 is the number of contigs. So that case would be 3, because you've got 3 contigs which count for 50% of the total size of the genome. So again, it's a useful indicator of genome sequencing quality, or quality of the assembly of your genome sequences. So those are just some metrics that you might come across. There are a few others as well, and there are various tools that you can use to assess the quality. And then obviously it becomes a decision as to whether or not you include that genome sequencing in your analysis. If it's of poor quality, you'd be worried about getting false positives or false negatives. So therefore you might decide to exclude that genome on the basis of quality. Assessment schemes for whole genome sequencing. So there's the PulseNet scheme. There was an ECDC work in this area as well. There are many, many approaches to this. So all I'd say to you is, if you're interested in this and you're looking at this, then just do a search on the current status of these various different schemes that are being used to try and standardize approaches between laboratories, and also to work towards accreditation of various steps within the process of genome sequencing, from the laboratory all the way through to the assembly and analysis as well. And developing standard operating procedures, even within your own organization, I think is a very good practice anyway, and ensuring that as part of that, you have quality control considered. And this, in order to be able to compare, it becomes particularly important if you're dealing with multi-jurisdictional events. So if you're crossing borders, having some kind of standardization, accreditation, harmonization also helps when you're trying to infer causes of events that are occurring across borders. And obviously, this needs good governance and management of the data. And this also feeds into things like the importance of sensitivity, whether it's commercial sensitivity, patient sensitivity, government level trade sensitivities, all of these things need careful governance and management in order to make sure that the whole genome sequencing data is being used for decision making effectively, but without resulting in severe adverse consequences either for a country's trade, for global trade, for a company, or for the well-being of consumers. And it's often a trade-off between all of those different competing demands as to how the data would be used. And the last point I'm going to make there is around access to whole genome sequencing in low resource settings. So, and this has become a real issue. I mentioned about these, it's the third time I've shown this slide. The international pathogen sequencing databases are heavily biased towards high income countries. And this is an example from PubMLST, another source of whole genomes. Only, for example, 0.4% of those genomes are from, this is of Campylobacter, are from Africa. The vast majority are from North America and from Europe. And this is an issue that really needs to be addressed. And some of the work that most recent work I've been doing, I've been in countries that, like in African countries, with, for example, Ethiopia, Kenya, and Tanzania, trying to increase the number of genome sequences and the use of whole genome sequences in those environments. And our recent publications on Ethiopia, if you're interested, might be of interest because they really illustrate how you can use whole genome sequencing even in extremely challenging environments. In Ethiopia, our study was done over a year during COVID and in a war zone during major environmental climate events or weather events as well. So there's a paper that describes the design of that study and how it was conducted under very, very challenging situations. But it is an issue, and we need to address that issue of the bias. If you're developing genome sequencing from scratch, this is a good paper, which really helps. This is one of the things that skills needed to develop and translate it. So if you're an organization where you're wanting to use whole genome sequencing technology, it's worth knowing what expertise as well as what physical resources you need to be able to do that. And that just table there illustrates the kind of expertise that you'd need. And again, this is the WHO 2018 and also the Listeria ECD paper, which describes, the question was asked around the challenges, and the challenges are, there's the technical and scientific challenges, but there's also cultural and organizational challenges that need to be borne in mind if you're planning on setting up and using whole genome sequencing routinely as part of your decision-making process. So just to finish, a couple of slides to finish with. Future challenges. I mentioned whole genome sequencing in low and middle resource settings is still a challenge. Clear guidance, governance and use around interpretation of the genome sequencing data. The need, as I mentioned, for these standardization, accreditation, harmonization, and this is right the way from sample collection all the way through to the interpretation and decision making based on whole genome sequence data. The importance, particularly around that when you've got multi-jurisdictional events, the need to develop standard operating procedures and document quality control methods and governance and management. And lastly, just there, I've talked primarily about whole genome sequencing where you have isolates from bacterial cultures that you're sequencing. What the future is around these culture independent methods. And this is something maybe you might want to have another webinar in a couple of years' time, because the field is changing so rapidly around the application of culture-independent methods, metagenomic methods, around pathogen surveillance. This is a Nature Microbiology one, which talks about harmonized pathogen surveillance in general, but it includes foodborne surveillance using metagenomic approaches. So no time to go into that today, but it is a future area to look into. So the final slide. So whole genome sequencing. I hope I've convinced you, and those of you who are already working in this, and maybe I've not told you anything new, but convinced you that it is certainly a worthwhile endeavor. It's something that I've been involved with extensively now for a couple of decades, and the development of these approaches and the application in New Zealand, but also internationally as well. not just for food safety, but also for other pathogens. How it scales from global to highly local, from global transmission to food processing environments, how we can use it to investigate outbreaks, how you can use it to identify the source of sporadic cases, and also for looking at things like virulence and antimicrobial resistance. And the tools that we use are developing alongside the technology for visualizing and using that. It's very useful, these tools, for working with government decision makers. If you can demonstrate using really nice visualization tools what's going on, it often has a higher impact. And again, just those challenges with standardization, harmonization, governance, and legal issues. And finally, just that issue, just to be aware of the emerging technologies around metagenomics and metagenomics related approaches. And obviously what I've described, there are a whole number of applications there and a huge number of people. This is a collaborative thing, and this is really important. It's One Health in action, but it's also highly collaborative. You need people with multiple areas of expertise working together to be able to use these tools most effectively, often in international settings as well as local settings, and it needs resourcing. A lot of the tools are still under development, but in the not too distant future, many of these will be routinely used in the food industry and at government levels as well for the benefit of food safety. I'll just stop there, and we can take any final questions that you might have.
Okay, Prof. Ranch. There is one question from YouTube channel of our department. The question coming from Saskia from UP University Indonesia. She said that topic given by you is very interesting. The question is specific for salmonella in beef, chicken, and egg. Saskia would like to hear more from your insight on how we can mitigate the source of foodborne disease in case typically with identifying is it from beef, chicken, or egg.
Yeah, that's where that approach to source attribution, and in particular how it applies to the reservoir host. So if you can use genome sequencing to say, given your human cases of salmonellosis, if you genome sequence a set of human cases and you also have isolates from animal reservoirs and from food sources, then you can use the kind of tools that I've mentioned to help refine your understanding of where those human infections are coming from. And you may be able to say, well, based on the types that we're getting from human cases, that maybe 50% of them are coming from cattle and 20% are coming from sheep or from poultry. Then you might be able to say, well, where within the poultry sector are they coming from? Are they coming from the egg layers or are they coming from the meat supply? And then that requires you to sample from those sources and then use the tools that I've described there to help you say, well, 10% are coming from eggs and maybe 80% are coming from meat. Then that helps you target the industry that is most responsible for those outbreaks. And that's where you can then do more detailed investigations. But backing up from that, we've used these tools to help the first question. So if you have a new strain of a pathogen that comes into your country, often one of the first questions that regulators want to know is, how long has it been here? And the reason why you ask that question is it may determine what strategy you subsequently use. So and we've used this in a number of occasions now, the salmonella example that I referred to earlier, the Salmonella enteritidis, we were able to show that it had arrived in New Zealand just prior to the major outbreak in the restaurant. If it had been in New Zealand for, if our analysis had shown it had been in New Zealand for 20 years, then an eradication program may not even be feasible. You may then go for suppression or mitigation. If it's only just arrived, then any further analysis which shows it's limited transmission within the food sector, then an eradication or elimination program might be more feasible than a suppression or mitigation program. So it can help you determine the strategy right at the beginning, but then also inform where you target your efforts and what interventions you might want to use to control, in that case, salmonella. And the enteritis example is a very good one from New Zealand, but there are many other examples as well.
Okay, thank you, Prof. Raj. There is one more question, but the question not one more actually, five more from Bu Sri Surati. Bu Sri Surati is from our governmental body, from our regulator PPOMN. So I will read all of the question, but I think you can prefer answer it in short time because we have limited time. Okay.
Yeah.
Bu Sri Surati said that thank you, Prof, for your very impressive sharing. Now, Bu Sri are developing a method using NGS to detect foodborne disease causative agents. In this case, Bu Sri used the metagenomic 16S rRNA and metagenomic whole genome sequencing approach. Technically, when validating the method, what validation parameter that Bu Sri need to be ensured? And what is the status of the test that used NGS? Is it only for screening and must be continued with another confirmatory test? Or can the result be reported directly from the NGS data? And the fourth question, for AMR analysis, what is the status of NGS analysis? When should NGS analysis be used? When there are other phenotype-based test methods? And the last question, in conducting analysis using NGS, are there any international guidelines or standards for conducting validation as well as bioinformatic analysis? Maybe you can answer the last one, and I will mention the next, the other question.
Yeah.
The last question
Yeah.
is, in conducting analysis using NGS, are there any international guidelines or standards for conducting validation and
Yeah, so
bioinformatics?
those reports that I mentioned in that last session, so there are some good examples there. The WHO documents and the ECDC around development of accreditation for listeria. So there are some really good examples based from the European Centre for Disease Control, from some of the WHO publications. So I'd really refer to those. So for how they're working, and maybe the previous one on AMR, I do have given similar webinars on the application of whole genome sequencing for AMR as well, and I did one for the Food Standards Authority Agency of New Zealand and Australia and New Zealand, and there for the phenotypic testing, if you compare phenotype with genotype, in the early days there wasn't such a good correlation between the two. But the more we know and the more genomes we have sequenced, the more genomes where we also have phenotype data, we can refine our understanding of the relationship between those two. And it's getting more and more accurate. And there are now also AI, artificial intelligence-based tools which can infer phenotypic resistance from novel mutations in the genome as well. So that technology and the tools, that there's one particular one called AMR Finder Plus, which has emerged as the kind of dominant tool that people are using now for inferring antimicrobial resistance from genome sequences. And fewer people are doing confirmatory tests in the laboratory, such as antimicrobial susceptibility testing. So it's getting better and better, the correlation between genotype and phenotype. So that's an important area to look at. Probably that's all the time I have. There are very other important questions, but I'm happy for my email to be shared if people would like to contact me, and obviously I can make a copy of my presentation available, because there are a number of references in that that people might want to follow up on.
Yeah. Okay. Thank you, Prof. Okay, because our limited duration, I think for the other question from Bu Sri and from the other questioner, I think we can answer it indirectly with another communication media, via email. Please send to our email from all of you from the floor. We have email in skpn@ipb.aca.id. Yeah. Okay, I think that's all for our segment. On behalf of the organizing committee and all participants, I would like to extend our deepest appreciation to you, Prof. Nigel, Prof. Friends, yeah, because very insightful presentation that you have taught to all of us. Yeah, thank you so much, Prof. And we also thank all participants for your active engagement for all of the questions. Yeah, and this webinar not only highlights the promises of NGS and WGS technologies, but also reinforce the importance of collaboration and standardization in safeguarding global food system, especially in Indonesian food system. Okay, we hope today's session has enriched our understanding and inspired our next or further discussion and research. Thank you again, and we wish you all meaningful World Food Safety Day in 2025. I will give the screen come back to the committee. Thank you.
Okay, thank you so much, Dr. Valerina and Professor French, who have shared expertise and insights on the latest advancement in the food safety research. And now, actually, we would like to share the token appreciation, a certificate, and I would like to invite Professor Eko and Professor Rati to deliver the certificate, and will be here shortly.
Eko is there. Yeah.
Oh, okay. So, yeah, so it's our pleasure to present the certificate of appreciation to Professor Nigel Friends as a speaker of today's seminar. Thank you very much for for sharing your expertise and insight with us today. And once again, thank you, and looking forward to continued collaboration with you in the near future. So with this, so we would like to present the certificate of appreciation to Professor Nigel Friends. Once again, thank you, Professor Nigel Friends.
Thank you very much. Thank you. Appreciate it.
Thank you very much, Prof. Eko and Professor French. Please stay on the spotlight. And I would like to invite also Prof. Rati to share the appreciation to the moderator, Dr. Valerina. Thank you.
Bu Valel, Dr. Valerina, thank you so much for helping and moderating the discussion so that it went very well. And we appreciate all of the attendance, and of course, especially for Professor French. Thank you.
Thank you so much, Prof. Rati, and thank you, Dr. Valerina. And now we are in the photo session, and we invite you to on your camera, and the committee will take a photo. Okay. So we can take a picture. There are like 5 slots. So in 3, 2, 1, cheers. Okay, page 2, 3, 2, 1. Thank you. Page 3. Page 4. Page 5. 6. Okay. So thank you so much to the participants. And we are in the closing of this event. And sorry for longer the time. So as we reach the conclusion today, I want to express my deepest gratitude and appreciation to everyone who has participated. Your presence and engagement today contribute to the global effort in ensuring the food safety system. So we hope that the webinar will share the valuable insight to you and inspire new collaboration in the field of food safety. Until we meet again, stay informed, stay engaged, and keep making a difference. Thank you.
Thank you, Nigel.
Thank you. Bye.
Appreciate it.
Bye.
Thank you
Thank you
Very
all.
much.
Thank
Have
you,
a
everyone.
good day,
Thank you,
everyone.
Bye, everyone.
Terima kasih Bu Rati.
halo Bu Dekan, selamat. Terima kasih Bu
Oh, ada Bu
Dekan.
Dekan ternyata di sini.
Ya, selamat ya
Izin,
Bu.
Bu. Izin, Bu.
Very proud of you.
Thank you.
Terima kasih Mas Asep, keren.
Ya,
Bu
terima
Farel as
kasih,
always. Terima
Prof.
kasih
Rati.
untuk support Pak Eko. Thank you
Sama-sama,
everyone.
sukses semua. Selamat Bu Rati, Valel, Formakepa, dan semuanya.
Heavy Pak, heavy science hari ini.
Ya, we need time to
Oke.
digest slowly.